reliable is it? SARS-CoV-2, like many other viruses, contains genetic material called RNA. RNA fragments are so small, they're difficult to detect. The PCR test doubles the fragments in what are called cycle thresholds and keeps doubling them until they have enough genetic material to identify. Most labs go up to 38 or 40 cycles, which means an amplification of 1 trillion times. But is that too much amplification? Because while live virus is present in people who are actually sick, traces of dead virus are present in people who used to be sick, even months later, or in people who were exposed to a virus, but their body cleared it and they never got sick at all. They're not infectious. Genuinely sick people get a positive test in relatively few cycles because they have a high viral load. But the samples of people who are recovered or exposed have to be magnified billions of times to get a positive result. That's why scientists are recommending not testing over 30 cycle thresholds. So what would happen to case numbers if labs cut back to 30? At one New York lab, it resulted in a 63% case reduction. When a Massachusetts lab did the same, almost 90% of their cases disappeared. And that's not the only way that PCR tests can go wrong. They can also misidentify remnants from the other 6 human coronavirus types as SARS-CoV-2.
And in fact, if you're testing in a population that doesn't have very much COVID, you'll get false positives almost half the time. The person actually doesn't have COVID, they have something else, they may have nothing.
In 2007, PCR tests caused a pseudo-epidemic for whooping cough at a hospital in New Hampshire. 100% of the results were false positives.
Because if you can amplify one single molecule up to something that you can really measure, which PCR can do, then there's just very few molecules that you don't have at least one single one of them in your body.
This is a delicate test that's supposed to be done by highly skilled technicians in a sterile lab, yet it's currently being done on a massive, expedited scale with hastily trained technicians working on huge numbers of tests. So the next time you see case numbers, ask yourself how many